Text: Office of Dietary Supplements Seminar. ods.od.nih.gov. Logo: National Institutes of Health Office of Dietary Supplements.
Okay, I am going to go ahead and get us started. Welcome everyone to today's NIH Office of Dietary Supplements seminar. I'm Barbara Sorkin. In addition to organizing ODS's seminars, I co-direct the NIH's Consortium Advancing Research on Botanicals and Other Natural Products, our carbon program. Before I introduce today's speaker, a few brief announcements. First, the coming attractions. Our next ODS webinar on February 28th will be given by Dr. June Round from the University of Utah. That will also be at 11 a.m. Eastern Time. And I believe Dr. Round is going to be talking about immune system, microbiome interactions, and health. If you'd like to join us for that seminar and you're not already on our seminar mailing list and would like to be, please see the chat for information for an email address where you can ask to be added to that list. It will be there shortly.
Text: ODS event guidelines. https://ods.od.nih.gov/about/ods_event_guidelines.aspx. Attendees are expected to uphold the professional and educational purposes of meetings by respecting the rights, views, privacy, safety, and dignity of all people in their speech, postings, and other actions. Keep all comments relevant to the event topic. Refrain from engaging in harassing speech or other inappropriate behavior. Posting insulting, obscene, discriminatory, or threatening comments. Disrupting the conversation, including with spam or repetitive posts. If you see or experience a violation of these guidelines, please contact ODS at ods@nih.gov or send a private message to the meeting host. Participants who engage in inappropriate behavior will be given a warning from event organizers, asked to stop the behavior and be expected to stop immediately. Participants who continue to display inappropriate behavior after being given such a warning will be removed from the event.
Next, some webinar logistics. In the interests of audio quality, we've muted you all on entry, and we ask that all of you except the speaker please stay muted until the final questions and answers. You have access to the chat, as I just pointed out. I encourage you to put any questions that you have for the speaker in the chat as they arise. If I see any that seem urgent, I will try to get Dave's attention to them midstream. Otherwise, we'll take as many as we can fit at the end of his presentation. Please also use the chat if you're having technical problems with the audio, the video, or with the closed captions, and one of us at the NIH end will try to help.
Text: Any opinions expressed by ODS seminar speakers or attendees do not necessarily reflect the views of the National Institutes of Health, the Department of Health and Human Services, or the United States government.
And now I have the great pleasure of introducing today's speaker. Dr. David Wishart is a distinguished university professor in the Departments of Biological Sciences and Computing Science at the University of Alberta in Edmonton. That's in Canada for those of you who are not familiar with our neighbor to the north. I'm honored to have Dave as one of the researchers driving the carbon program's Natural Product Magnetic Resonance Database project. His research interests are wide-ranging, covering metabolomics, analytical, computational, food, natural product and protein chemistry, molecular biology, and machine learning. Dave has developed a number of widely used techniques based on nuclear magnetic resonance spectroscopy and mass spectrometry to characterize the structure of molecules, both small and large, and he has led the Human Metabolome Project for nearly 20 years. The information from that HMP has been archived on a freely accessible web resource called the Human Metabolome Database, the HMDB. And Dave has more recently focused on using the same metabolomic methods developed for the HMDB to help characterize the chemistry of biological mixtures, other natural products, foods, beverages. This work has led to the creation of a number of additional popular databases freely available online, including, as I mentioned, the Natural Products Magnetic Resonance Database, FoodDB, Phenol Explorer, PhytoHub, and MarkerDB. And I'm sure Dave will do a better job of telling you about those databases and the ways that those can be used. So Dave, I'm going to hand the microphone over to you. You're not going to hear anything further from me until the end of your talk or the end of your time, whichever comes first, unless we're having trouble with your audio or visual, in which case I promise I will let you know. So I'm going to stop sharing and shut up.
Great. Thanks so much, Barbara. It's a real pleasure to be here. And I also want to thank you for the invitation to present today. So I guess this is the first virtual seminar of 2024, and really glad to be part of this. So the title is a little awkward, rather long, but I'm trying to blend together some of the things that Barbara talked about in terms of my research and interests. So we're going to be talking about metabolomics, dietary supplements, and then online databases, but how we can actually try and use those to make people healthier. And this is with regard to precision nutrition. I want to make sure I can get the... So first thing to do is maybe get everyone aligned with this idea of what metabolomics is. Probably most of you have heard of it, but just as a way of getting everyone at the same level, I often compare metabolomics to genomics, which I think everyone's more familiar with. So basically we use high throughput sequencing in genomics to do DNA sequencing, RNA sequencing, and we try and characterize genes. We look at things in biofluids like blood, or we might look at cells, tissues, or organisms. So we're looking at the genome in genomics. In metabolomics, we're looking at the metabolome. And we use a different set of technologies. So instead of DNA sequencers, we use things like mass spectrometry, nuclear magnetic resonance spectroscopy. We separate molecules by liquid chromatography or gas chromatography. And we're looking at things much, much smaller than DNA or RNA. Usually we're looking at molecules about 1,500 Daltons, but we'll look at fluids, we'll look at cells, we'll look at tissues, or we'll look at entire organisms. So that's I guess, the brief definition of metabolomics. It's also useful to sort of understand metabolomics in context. There's the omics pyramid, or the pyramid of life, as I call it. At the base is the genome. Genes code for proteins. Proteins then sort of manipulate metabolites to transform them, change them, break them down. And so study of the genome is genomics, the study of the proteome is proteomics, the study of metabolome is metabolomics. As you climb up the pyramid, you'll generally notice that some things are more heavily influenced by external factors. So the genome is and shouldn't be influenced by what you eat. Otherwise, we'd be in a lot of trouble. Typically, it is only lightly influenced by maybe exposures like radiation. The genome is barely influenced by the microbiome, and likewise, the genome is identical in basically every cell in your body. But as you climb up this pyramid, you'll find that there's progressively greater influence in diet, environment, the microbiome, and in physiology. And so by the time you reach the top at the metabolome, you'll find that the metabolome of the heart is very different than the metabolome or metabolism of the brain, which is very different than the metabolism of the skin, yet all of those have identical genomes. I've put a bird at the very top. It's actually a canary, and it's to remind me to tell you that really metabolites are like the canaries of the genome. So
Dave, could you project a little bit more, a little bit louder? People are having difficulty hearing.
Sure. So basically the canary of the genome concept is that metabolites are greatly amplified. So a single mutation at the DNA level can lead to about a 10,000-fold change in certain metabolites. So this makes metabolomics and metabolite measurement a really useful thing for monitoring what's going on at the genome, but also for understanding what's going on in the environment. So the metabolome is a great way of getting a readout of the phenome or the phenotype.
A graph appears titled, Technology and Sensitivity. The axes are labeled, number metabolites detected and sensitivity or LOD.
Now in metabolomics, we use a bunch of different technologies. We'll use NMR spectroscopy, gas chromatography, mass spectrometry, liquid chromatography, mass spectrometry. This sort of plots out the sensitivity that we have with different techniques. So NMR is the least sensitive. LCMS is the most sensitive. So we can measure down into the sub-nanomolar level with LCMS. With NMR, we're usually at the 10s or 20 micromolar, and GCMS is in between. With NMR, we can measure most compounds and understand what they are. We identify them. So we call those the known unknowns. In LCMS, where we might measure up to 10,000 different features, a lot of them, probably about 90% of them, are unknown. We're still trying to figure them out. And this is where metabolomics is trailing rather badly behind genomics, where in fact, most of the time we know what all the genes are. And even in proteomics, where we know most of what the proteins are. In metabolomics, we're still trying to figure out a large number of them. And that's one of the interesting challenges, and that's one in the area I've been working in a long time.
Text, 2 routes to metabolomics.
The other thing to remember in metabolomics is we have 2 different approaches. One is called targeted metabolomics, where we choose a set of anywhere from 100 to 1,000 specific metabolites, where we have references and actual compounds in our hands, and we measure them very specifically, and we quantify them. Untargeted metabolomics picks up from where proteomics was, which is we don't know what we're going to see. We measure as much as we can detect. We see what are called features. And typically untargeted metabolomics it doesn't allow us to quantify things. It does allow us to discover things or identify potentially new points, and then we go back to use targeted quantitative metabolomics to figure out what these compounds really might be. About 80% of the papers in metabolomics are in the untargeted domain, and about 20% are in the targeted domain. My lab focuses mostly on targeted metabolomics because I'm interested in quantification, and I'm interested in translating the findings into medical applications. And that's targeted metabolomics is the only way you can do that.
Text, targeted metabolomics with NMR, www.magmet.ca.
So Barbara mentioned I work in the area of NMR and have for many, many years. We can do metabolomics or targeted metabolomics with NMR spectroscopy. And how we do that is sort of shown on the left side, where you could imagine a mixture shown in blue. And if you look at the red, green, and purple spectra, which correspond to 3 different compounds, A, B, and C, if you sort of mentally sum those spectra together, you can see how we get the blue spectrum. Now, our challenge in metabolomics is to do the reverse, to deconvolute that mixture and identify those individual compounds or individual spectra. In the middle, I show a program we've developed called MagMet, or Magnetic Resonance for Metabolomics, that does this automatically. And you can see some of the fitting that it does, and then there's a list of a couple of the compounds on the far right. You can see sort of the difficulty in terms of the number of peaks that we have to deconvolve. So it's a very challenging computational problem, but it's largely been solved. And so now we can automatically analyze complex mixtures with NMR.
Text, non-destructive, simple sample preparation, fast, easy to perform, cheap. Fully automated data collection, data processing and compound identification of hydrophilic compounds, sugars, alcohols, amino acids, organic acids, etc. Detects and quantifies 55 to 60 serum metabolites, 75 fecal metabolites, 100 plus urine metabolites, 50 to 75 food and beverage compounds in less than 5 minutes.
So this is just showing some examples where we're analyzing things like serum, where we get 57 compounds, or fecal water, where we get about 72 compounds. And the computer can do this in about 5 to 6 minutes. NMR is really appealing because it's non-destructive, it's simple sample preparation, it's very fast, takes about 3 minutes to collect the spectrum. All the analyses can be fully automated, so you just run it while you're sleeping or run it in parallel. So the data collection, the data processing, and the compound identification is all done now automatically. It's mostly focused on hydrophilic compounds like sugars, organic acids, and alcohols. And depending on whether you're looking at biofluids in humans, you can get between 50 and 100 compounds. Or if you're looking at foods and beverages or supplements, between 50 and 75 components. As I said, it's really cheap. With the center that I operate in Canada, it's about $10 per sample. Now, the other method that I highlighted before was liquid chromatography mass spectrometry, and I indicated that was the most sensitive technique. Now, this is a picture illustrating the approach that we use to do targeted LC-MS metabolomics. So in this example, we're taking, say, biofluids. We put them into 96-well plates. We process them with certain derivatization methods. These have carbon-13 labels on them, and this allows us to do quantification. We run these 96-well plates. It can take 2 to 3 days to look at 90 or more samples. And then we run them through various data processing tools. The software we've developed is called LC AutoFit, and it's a lot like that NMR magnet software. So it does that deconvolution, and it figures out the compounds based on the reference spectra, the retention time, the MRM transitions, and it does the integration to get the quantification, and then it spits out a report.
Text: what's possible with LC-MS/MS? 3 tables appear noted as big, bigger, and biggest.
So we've developed a bunch of assays, and then we've converted those assays into kits. We use the kits within my lab, but we've also distributed the kits to other labs around the world. And we can work on, one version is called the Prime. It measures about 180 compounds and compound ratios. One that's quite popular is one called Mega. It measures about 650 compounds in about 20 different categories or classes. And then you can calculate a whole bunch of ratios or sums. And this is the nice thing you can do with quantitation. Because you can accurately measure quantities, the ratios mean something or sums mean something. If you aren't quantifying, then you can't add things up or you can't calculate ratios properly. More recently, we've developed a new assay that measures about 1,400 compounds. It's called the Giga. And then from that, you can get about 450 ratios. So we're actually talking about 1,800 values that you can typically measure from LC-MS. So that's, about 20 to 30 times more than what we can measure by NMR. It's a little more complicated. We have to use isotopic labels, so that costs us some money, but it's still pretty cheap, between $40 and $75 to get these sort of kits and to run them in a lab. The software, as I mentioned, LC AutoFit does all of this automatically, and typically to process an entire plate, which might have 90 samples, and you're measuring 1,400 compounds, can all be done in about 6 or 7 minutes.
Text: measuring our food, natural products.
So I've told you a little bit about metabolomics, and I'm going to dive into, what do we use it for? So I've been using it, and many other people have been using it for a long time to measure food. But historically, for the last 100 years, we've been using other techniques to identify or measure components in food. So if you look at a food label on your cereal box or on anything else that you get with food labeling, you'll see maybe about a dozen things listed. You might see protein, fiber, sugar, fat. These are using methods that were developed in some cases more than 100 years ago. The gel doll method is about, I don't know, 80 years old. Fiber has been around, measurements have been refined. Ash has been measured historically using just weighing burned out stuff, but now people use FTIR. The SOSET method for, or SOXET method for fat. Again, ancient chemistry. More recently, people started measuring minerals like sodium and calcium. So that's usually done by atomic absorption spectroscopy, and then vitamins by LCMS. So as I said, standard methods for measuring food might give you between 10 and 20 compounds. But with metabolomics, we can measure actually thousands of values. So the approach in food metabolomics is take your food samples, extract them, take those extracts and put them into NMR spectrometers, LC or GCMS instruments, and then do a variety of data analysis techniques. I won't go into all the details, but I've already mentioned some of the software tools that we do this, and you could do both targeted and untargeted. But targeted is more valuable because you actually get concentrations, and those are things that are pretty relevant in terms of nutrition and health.
Text, example food constituents.
So when we do food metabolomics, we can look at things like tea and meat and eggs and wine and soy. And some of these compounds might be familiar, but certainly none of them are listed on any food label. And a lot of these actually are really, really important. Some of them can be used to determine if you are eating certain foods, but some of them also have impact on your health. Lutein in eggs is very important for preventing macular degeneration. Daidzein and other soy products are broken down in equal, which has very important health effects, positive health effects. Alkyl resorcinols are vital to the health impacts of grain, along with betaine, which is also highly abundant in grains and is known to have very positive impact in health. So these are food constituents that are never measured by the standard techniques, but are routinely measured and measurable by the methods I just talked about in metabolomics. We've had a bit of fun over the last few years using NMR to measure wine and beer. And this is pretty easy to do.
Text, automated wine analysis.
The fluids in wine and beer are relatively simple. This is a spectrum of what you'll see in wine. You'll see ethanol, of course, but you'll also see glycerol, malic acid, tartaric acid, and in total about 90 compounds can be read out and quantitatively measured from almost any given wine sample.
A table appears titled, Oyster Base Sauvignon Blanc Metabolites, Mg/L.
And some level, it's intriguing to see what's there. It's also sometimes scary to see what's in wine. There's not just ethanol, but there's methanol, there's propanol, there's butanol. Some of these obviously are toxic, and there are regulations about how much can be in some of these wine samples. Others help us explain some of the aromas and scent and taste for wine. And in fact, it's really intriguing to see just how complex chemically wine and beer are, and how these things contribute to the mouthfeel, the taste and aroma that we have. You can do this with milk. We've done this again with NMR.
Text, automated milk analysis.
And we can detect about 65 compounds in the average commercial milk sample. And this again can be done in about 4 to 5 minutes in terms of both the calculation and the spectral collection time. So this is, probably about 10 times more compounds that is what are normally measured or reported in your standard store-bought milk. Now, NMR, as I said, is not the most sensitive technique, and it's slowly being usurped by LCMS. This is an assay, and it's now been converted to a kit that we use to measure foods. And it lists about the 20 different major categories that we detect. It uses the workflow that I described before, which had those derivatization methods, LC autofit. And you can see that we're measuring lipids and lipid-like molecules, we're measuring organic acids, amino acids, derivatives, biogenic amines, vitamins, and we can also measure metals using something called ICP-MS.
Text, example kits, and essays. A table appears with the title, Animal Food Panel Part 1, 84 compounds.
So in terms of the types of compounds and the actual details, here's a list of some of them. They're colored according to some of their roles. You can see some of the vitamins. You can see some of the organic and fatty acids. You can see some of the phenolic acids.
More tables slide in.
You can see some of the acyl carnitines, the lipids, the sphingolipids, more of them, ceramides, cholesterol esters, diacylglycerols, triacylglycerols, more triacylglycerols. And then here are some of the metals. So it's lots and lots of compounds, more than what's easily displayable on a single screen. But this is the scale that's now possible with metabolomics, and all of these can be quantified with an error of maybe about 10% down to sub-micromolar or nanomolar levels. So that was an example of animal foods. This is an example of when we're analyzing fruits and vegetables and the types of compounds we can get. And you can see again some of the organic acids, the vitamins, amino acids, other molecules that might include the polyphenols, the terpenoids, sugar alcohols. You can also detect herbicides and pesticides, and again, the metals. So depending on the type of food and the source origin, we can customize or adapt the assay or the kit to measure specific compound sets. So we've been starting to do this on our own, looking at different foods that we get in Canada.
Text, metabolomics of beetroot. A bar chart shows levels of various compounds.
So we grow lots of beets here, including sugar beets. But this is an example of the number of compounds that we were able to detect and quantify using LCMS, which is on the right in blue, ICP-MS, which is the green one, GCMS, which is the purple, NMR, which is the red. And those are some examples of some of the instruments that we use in my lab and the number of quantified compounds. So we haven't done just beets, we've done zucchini and cabbage and raspberries, eggplant and broccoli. And using multiple assays, we can get hundreds of compounds identified and quantified.
Text, measuring our diets and supplement intake. Food frequency questionnaires. 24-hour recalls. Weighed food records. Dietary histories. Food supplement diaries. Metabolic research kitchens. Photographic records. Metabolomics.
Now that's food. And knowing what we eat is obviously important, and we can do the same thing with supplements or supplement mixtures. But I think an equally important thing, especially with health, is how much are we taking in of these things? And are we processing those in our bodies in the right way, or are we getting too much or too little of these things? And so in the world of dietary science, people use a variety of questionnaires like food frequency questionnaires and recalls and food records and histories and supplement diaries. And sometimes they'll put people into kitchens and closed rooms. And sometimes they'll ask people to take photographs with their cell phones. But again, more and more are realizing that you can use metabolomics to get the answers for most of these things. In many cases, the answers are more accurate than these questionnaires or photographic records. So when we do food intake metabolomics, we start with food, but we get people to eat the food. And from that, we can measure from either the urine or biofluids that we collect, like blood, and then do the same chemical analysis and the same data analysis to figure out what people have eaten. So one of the things we have to remember when people eat food is these things get transformed. They go into our mouth, and amylases knock off some of the sugars. They go into the bloodstream, which then go into the liver. There's phase 1 and phase 2 transformations. These things then go past to the kidneys or the gut. The gut will use microbes to do some transformations, but some of those go back in the blood, back in the kidney. Some of them go into the urine, and so on. And you can get all kinds of combinations of different reactions for a given starting compound, like isoquercetin, which you'll get in apples or onions, and it'll be transformed into about 250 different compounds through all these different compartments and processes.
Text, example food intake biomarkers.
So I took an example of some of the foods we looked at, and in light blue are examples of some of the food compounds that we measure. But these food compounds are very quickly decomposed in our body into things like tea converts to hippuric acid, meat's converted to methylhistidine, lecithin in eggs is converted to trimethylamine oxide. Wine, the ethanol is converted to ethyl glucuronide. Soy, the daidzein is converted to equal, and so on. And I've marked in different colors indicating what these compounds are the products of. Some are a result of just straight catabolism, some are phase 2 metabolism, some are phase 1, some are microbial, or some are phase 1 microbial co-metabolites. So it gets complicated, but some of these end products actually are great markers for telling us if someone had eggs, or if someone had soy, or if someone ate onions, or if someone drank coffee. And these things can only be measured through metabolomics. And by measuring the amount, we can actually determine how much of any given food someone has eaten. So as a consequence of that, we've started developing food intake kits and assays. And this is one which measures about 300 compounds, which are relevant to food intake. And they've again broken down into different components. Some are formally called food intake markers, but others have certain classes and chemical categories, but we also know which food they're associated with. Now, you can do this not only for foods, you can do it for supplements, dietary supplements. And again, the same process is have someone take the supplements, get the biofluids or extracts, do the same sort of chemical analysis and data analysis. And in this case, this is a list of examples of the types of supplements that are popular these days. So there's multivitamins, there's beet extracts, creatine, probiotics, fish oil, turmeric, melatonin, antioxidants. And what I've marked in red are some of the compounds that we can typically and routinely see. So if someone takes multivitamins, we see increases in both blood, urine, and feces. If people take beet extracts, we'll see these three compounds, betaine, dimethylglycine, and sarcosine, elevated, but we'll also see homocysteine drop. Someone takes creatine, we'll see both increased creatinine and phosphocreatine. Probiotics, depending on the probiotics, we can see increased short-chain fatty acids, increased lactate, but a drop in indole derivatives and a drop in cresol derivatives, and so on. Now, all of these compounds may not be familiar to you, but they are measurable through metabolomics. And in this way, we can track whether someone is taking supplements or whether the supplements are being processed by their body properly, or whether they're taking too much or too little.
Text, LCMS/MS supplement intake analysis.
So the assay we run is very similar to the one that we do with food. And that's because a lot of supplements are derived from foods. And again, this measures about 300 different compounds and quantifies them. Now, the other thing to remember is that, we can measure what's going into your body. We can also measure what's, outside the body in terms of food and supplements. But then how do you measure their impact? So I highlighted if you take, a beet extract, it is known to reduce homocysteine, and homocysteine is a bad compound. How do we measure that? Now, normally we don't measure homocysteine in the supplement intake, but we do measure it in this one, the assay, which has 1,400 metabolites, and that's called the GIGA assay. And this lists the number of compounds and the different categories. And these are all tied to various pathways, various health effects. Some of them are known health markers. And so we can take blood and serum and not only measure what you've taken, that's one assay, but what its impact has been metabolically on you, your microbiome, and your general physiology. So just to summarize, I've talked about a whole bunch of different assays that we work on, that we measure, and that many other people are using now. And there's NMR tools to measure food and beverage, or to measure the impact on your blood or serum or stool samples, typically between 50 and 100 compounds. with LCMS, we can measure about 700 food compounds. We can measure food intake or supplement intake with assays that measure about 300 compounds. And then in terms of measuring their impact on your physiology, your metabolism, your health, we can measure about 1,400. Now, using the techniques like NMR and mass spec, we can typically measure between 20 and perhaps 100 samples a day, and that the price or cost is anywhere from $10 a sample for NMR up to $75 a sample for the large scale assay of 1,400 compounds. Now, I think the reason why I'm highlighting that is that a lot of people think that metabolomics is well outside of their reach, either that they could put into a large scale study or that it's too slow. And what I'm really trying to say is, no, it's actually very fast. This is just on a single instrument, up to 100 samples a day. And these prices are certainly well below what you typically would pay, say, for an RNA-seq analysis or a proteomics analysis or a transcriptomics analysis. So it's a very cheap process. It's many times cheaper than doing food analysis using the formal methods. It's obviously many times cheaper than what you would do if you tried to do clinical tests on all of the compounds here. The clinical tests, if you did all of them separately, would cost somewhere around $10,000 per sample and take about a month. So the question is, what can we do with all these sorts of tools that can measure, not only foods, but blood and serum and stool samples, and measure supplements and supplement intake and their impact?
Text, data mining, and experimental integration.
Well, what we started doing is we started collecting information on all of these foods, and we started measuring samples on people. But we also started looking in the literature because people are publishing on this stuff and have published for a long, long time. And so we started creating databases to help not only ourselves interpret the data, but other groups interpret them. And this is something that Barbara mentioned before about our focus on making databases. So for the last 20 years, this is kind of what it looked like for our database effort.
An image depicts students studying in a collegiate library.
I would hire dozens of students, and they would go into the library and they would read articles and enter that information onto their computers, and we would build databases. And we still do that today.
Text, newer approach, text information extraction using machine learning.
More recently, we've started using machine learning, and we've developed some tools like PolySearch, which allows us to do text mining across PubMed abstracts and many other databases to extract triples. Given X, find Y, or what does Y do? This is something that's important in information extraction. More recently, we've started using ChatGPT and other large language models to parse out text and also extract triples or relationships between subjects and objects. And so this is a major focus of our effort because I can't afford to hire dozens of students and staff anymore to do all of that database research. And so increasingly we're having to use AI and machine learning.
Text, the drug database, https://go.drugbank.com.
So over the course of the last 20 years, we started building databases. One database that some of you might have heard of is called Drug Bank. It covers all approved drugs, it covers supplements, it has detailed compound description, health effects, mechanisms of action, absorption, distribution, metabolism, pharmacokinetic data, covers FDA small molecule drugs, the biologics, and then a lot of nutraceuticals and supplements. It also has information on experimental drugs, their metabolism, their spectra. What most people use it for is its information on the targets and the pathways and how these drugs work and which targets they get or hit. And a lot of things have been repurposed because of Drug Bank to be applied to new applications. So this is a database. It's publicly available and lots of people use it for different purposes.
Text, literature mining, and accept. Metabolomics, Cannabis DB. https://cannabisdatabase.ca.
More recently, we've been looking at cannabis. In Canada, cannabis is legal, and obviously in a few states it's also legal. And there's obviously concern about what is in cannabis and how does it work. So we've been assembling this information. We've been analyzing cannabis. We've been putting together detailed descriptions of about 6,000 different cannabis compounds. We've gathered information on the health effects and presumptive targets and pathways. And we've used these metabolomics assays that I've just talked about to measure a number of the compounds that are in different cannabis samples. Just like Drug Bank, we've also got information about the protein targets. The paper describing this was just published, and the database is online now.
Text, literature mining, and accept. Metabolomics, the milk composition database. www.mcdb.ca.
We've taken the studies we did on milk. I showed you some NMR spectra of milk. Alberta is a very big dairy producer, large agriculture community. And we've done this, applied NMR, GCMS, LCMS, ICP-MS to measure hundreds of compounds in milk. We've taken literature and collected about 2,300 compounds. We've got concentration data, and the databases allow you to do structure, spectral, and text searches, and you've got very detailed compound descriptions about what these things are doing in milk or why they should or shouldn't be in there.
Text, literature mining and in-house analysis. The Food Constituent Database FoodDB. https://www.foodb.ca.
Another major effort has been a database called FoodDB. So this isn't just about milk, it's about every food, or at least the 730 or so raw or lightly processed foods. So these are called foundational foods. So these are the foods that allow you to make all the dishes that are interesting, to make pizza or pad thai or something like that. We put in information about obviously their chemistry, but their flavor, aroma, color, and their human health effects. Again, we've been measuring lots of foods using the metabolomics method, so that information's in there, along with the literature data. And so we have millions of concentration values, tens of thousands of references. And for about 2% of the compounds, we have health effects. We're still trying to find more information about that. And then about 3% or 4% have information on their flavor and aroma attributes. So this database is still evolving. It's available and has been online for some time, and we're hoping to release the official version later this year with a publication.
Text, https://www.np-mrd.org.
Barbara mentioned our work in the area of natural products, and that grew quite naturally out of our work in foods, because a lot of foods have many, many natural products. But also people use natural products as medicinal applications and obviously in supplements. This database now has almost 281,000 compounds. Its focus is on NMR. So we have thousands of experimentally collected NMR data, most of it collected in my lab. We have simulated NMR spectra, where we've taken chemical shift assignments in the literature and generated spectra. And then we've used machine learning to predict the NMR spectra of most of the other compounds, and that's led to about 2.5 million predicted spectra. But this isn't just simply a list of natural products. It has the descriptions, it has their potential applications or utilities in health. And we're using some of the machine learning methods for data extraction to enrich this information. So we'd like to turn this natural products database into something just as rich and richly annotated as Drug Bank, where we have information about things like mechanism of action and protein targets and other things. So we built these databases that describe, what's in your food, what's in cannabis, what's in natural products or known natural products, what's in milk. We've done it for polyphenols in fruits and vegetables with Phenol Explorer, and those lists and databases go on and on. But we've also created databases that measure the impact of food and diet and supplements on human health.
Text, https://markerdb.ca.
One database is called MarkerDB. So this is a biomarker database. We're looking for, if you talk about markers like, glucose for diabetes or cholesterol for heart disease or phenylalanine for phenylketonuria. these are examples of biomarkers, and there's metabolite biomarkers, there's genetic biomarkers, there's protein biomarkers. And we've compiled this from the literature. Some of these markers we've also published and identified ourselves, and we put into the database. We include sensitivity, specificity. We've got protein, gene, metabolite, karyotype. In the example of metabolites, there's about 1,100 metabolite disease diet biomarkers, about 220 exposure biomarkers, 184 food intake biomarkers, and thousands of protein and gene biomarkers. So this one is something that quantitatively tells you what are these things doing and how are they indicative of either disease or impact or what you've consumed or been exposed to.
Text, https://www.hmdb.ca.
Barbara also mentioned the Human Metabolome Database. This is probably our largest database. It has about 250,000 human metabolites, and it's got metabolites that are endogenous. It has microbial metabolites that are produced in your gut. It includes the dietary compounds that come from your food. And we have information on their structures and names and physical properties. And we've measured and collected spectra on these and quantified. Many of the concentration values are measured in our lab. We've also generated thousands of pathways, millions of spectra, and data on hundreds of diseases. So this is a very, very rich resource that's used widely. In fact, the databases I've just been mentioning are accessed between 70 and 80 million times a year.
Text, https://mimdb.org.
Another new database is the Microbial Metabolome Database, and this is trying to link what our microbes in our gut produce and how the food we eat changes those metabolites, but also those microbes. So it has information about microbial origins, food origins, metabolites, as well as their effects on human health. Surprisingly, there's about 25,000 metabolites that seem to come from our microbiome. And there's about 2,000 species where we have sequence data. And then we've connected it to hundreds of diseases and mechanisms in this database. So this is one of our more complicated databases because it covers more than just metabolites. It covers genes, proteins, microbes, pathways, food, and food degradation and metabolism. So I've talked about metabolomics. I've talked about how we've used our tools in metabolomics and our machine learning and library resources to create databases. So how can you combine databases and metabolomics to advance human health and do something like precision nutrition? So precision nutrition, I think many of you are sort of familiar with it. Simply stated, it's getting the right nutrients, the right supplements, the right diet to the right person at the right time to optimize their health, increase their lifespan, improve their sense of well-being, or enhance their health span. So basically, people are assumed to have different responses to different foods. So certainly the best diet for one person may look very different for another. Now, precision nutrition, I think, is often regarded as more an aspirational thing than an actualized deliverable. But I'm going to try and show you that it's actually starting to have some impact and that using the tools I've talked about is helping that happen.
Text, best example of precision nutrition, treating IEMS with supplements.
And one example that a lot of people ignore in terms of precision nutrition and its success is the application of medical foods for inborn errors of metabolism. So this is an example where someone is diagnosed, usually by a metabolomic assay, because we do newborn screening and we measure blood spots and we identify people with PKU or people with MSUD or people with biotinidase deficiency. And once we've identified those people, we put them on a diet. And we give them things that might be high in biotin or high in carnitine or low in protein or low in branched chain amino acids or low in phenylalanine. And these diets quite literally save these people's lives. They're transformational. So this is one example where precision nutrition has worked and where metabolomics, coupled with obviously historic databases, allows clear interventions to be made and lives to be saved. If we expand it to the idea of precision nutrition for people without inborn errors of metabolism, we can think of trying to look at a group of people. We can measure their genome, the microbiome, the metabolome, And from that, we can start classifying people. And based on a few tricks I'll show you, then you can say, well, okay, these people have some similarity based on their either physiology or challenges or dietary imbalances. Will prescribe or suggest a keto diet, vegan diets, or certain supplements to correct that. Now, that correction often has to be done first through a detailed metabolomics test, and then trying to integrate some of the databases I've just talked about, like FoodDB or MindDB or HMDB, along with a variety of machine learning tools to interpret both the experimental data and that literature or computational data to come up with suggestions for nutrition or supplements or exercise or medications. So in many cases, it's a combination of metabolomics, databases, and either AI or machine learning. And these are some examples of companies that are doing that.
Logos, ILO, Viome, Molecular U Personalized Health, Day 2, Atlas, Sarvaris Aerospace, Zoe, HLI Human Longevity Incorporated, Whole Biome.
So they are doing the metabolomics. Sometimes they'll do microbiomics. Sometimes they'll combine it with proteomics. And then they use the databases. Most of them are the ones that I just talked about. Most of them are these freely available databases that we put out there that almost all of these companies now use to interpret the measurements that they make for their participants or patients.
Text, precision nutrition companies.
So targeted metabolomics is widely used. Microbiome or proteome tests, they'll look at these nutritional abnormalities, they'll look for food intake biomarkers, diet propensity. They'll use the machine learning, they'll use the databases like HMDB, MarkerDB, and MindDB, and that helps them inform and modify those diets to fix either nutrient excesses or deficits. And then they'll often have life coaches and continuous monitoring to help ensure some compliance. Many of them will do longitudinal profiling, where they might measure you over a couple of days or even a couple of weeks, or sometimes month to month and year to year to sort of optimize or make suggestions.
Text, impact of precision's nutrition. A line chart appear with several colored lines increasing across the graph. The axes are labeled health scores and baseline 100 days action plan.
So this is one example with one of these companies that used our databases and also was using some of our metabolomics kits and assays. And this is measured over the course of 100 days. And they were looking at 122 different people. And these are sort of the average scores that people had in terms of their dietary health, cognitive health, kidney health at the beginning, baseline. So their scores are below 60 and some below or near 30. And then they tracked these groups, and then this is their average value after that time and the average score that came after 100 days. And you can see that every one of these values improved. And this is because they were given both very specific dietary advice, but also some lifestyle. As it turned out, it was mostly diet, because most people didn't follow the lifestyle or exercise regimes. But this is an example of how not only metabolomics, but also the databases and then some of the machine learning tools that were implemented were making differences. And this is one example. There's several other companies, I won't mention them by name, but also use the same databases, the same assays we've just talked about, and they're getting very similar results over these 30, 60, and 100-day periods. So to conclude, I just want to emphasize that targeted metabolomics is a really powerful approach. It's really inexpensive, and it allows you to understand not only the composition of food, it can help you understand the composition of supplements, drugs. It can also help you understand how much has been consumed or not consumed. And more importantly, it can be used to assess the impact of those diets, drugs, and supplements on physiology or metabolism. Now, making use of modern information extraction methods, machine learning, AI, large language models, along with the old library approach, you can couple that with targeted metabolomics to create these comprehensive databases that cover whether it might be just the composition of food or cannabis or milk, or the markers that are relevant to disease, or the human metabolome, or the microbial metabolome in humans, and how they connect those compounds to diet and health. So that connection between health, metabolites, the literature, and what we can experimentally measure is key. So when you use targeted metabolomics and these online databases, as a number of those companies that I just highlighted are doing, they're starting to do precision nutrition. And so a nascent precision nutrition industry is really starting to emerge. And the results that I'm seeing, and some of them are starting to publish, are actually quite encouraging. And it's clear that improvements, measurable improvements in human health are possible through this guided, customized, individualized diet approach.
Text, acknowledgements. Logos, Alberta Innovates Biosolutions. The Metabolomics Innovation Center. National Institutes of Health, Genome Canada, CIHRIRSC, Innovations.ca Canada Foundation for Innovation, NSERC-CRSNG.
So with that, I'll wrap up. I want to thank my lab. There's too many people to mention by name, but this work has been done by dozens of people. And obviously the funders who've helped support the work over the last 15, 20 years. And thank you for listening.
The presentation closes, leaving just the speakers.
Wow, Dave, thank you. Fabulous. For someone who got his slides in late last night or early this morning. So I'm allowed to say that, right? We all do it. Okay, there are lots of questions already, and great questions. I'll put the questions about, can we get your papers at the end, and people can always email us at ods@nih.gov to ask for the references if they're having trouble finding them. But let me start. There are a few questions that I think are great questions, and I sort of had the same question, and you addressed it a little bit, but not really in your part about precision nutrition. And the question, of course, is you can do metabolomics on what goes in, and you can do metabolomics on the person. And how much variation do you see in terms of how what goes in changes the individual metabolomics? How much does that depend on how much variability is there in half-life of specific metabolites over the population? How much does it matter how long ago the person ate the whatever, ingested it?
No, I think that's a really good one. And that's an entire seminar on its own. So we can call these things biomarkers of food intake, or BFIs. We used to call them food intake biomarkers, but that comes out as a fib. So BFI is the term that we've used for the last few years. And this is a project that was started called Foodball in Europe, and has sort of been continued, I think, also as part of this All of Us Precision Nutrition Initiative. And there's another one that's also going on, where we're also sponsored by NIH. And this is to try and expand and understand these BFIs, get a larger list of them, and to understand that variability between humans. To qualify as a good BFI, you can't have a whole lot of variation. So there are examples like daidzein and soy and equal, where there's a huge amount of variation between people. One biomarker that stands out as really good is proline betaine, which is a marker of citrus consumption. And they've done study after study after study, and it's a compound that just isn't metabolized by humans, but it's found in citrus fruits. So it's inert. It's like metformin, just it doesn't get transformed. So it's a very good marker, and the correlations are about 0.7 to 0.8 in terms of proline betaine and the reported consumption of citrus. And we think the error is in the reported consumption, not in the actual measurement. So it may have almost a perfect correlation in citrus. So there's about 40 markers that sort of pass that level, but these are only good for about 12 to 24 hours. If you wanted to have markers that are measuring, what was your consumption of alcohol 6 months ago, there are certain lipid ethanol derivatives that can be used to measure that. We can also measure hair. And in that case, we've done studies of pregnant mothers And we can go back 8 to 9 months with hair, slice it up and identify food consumption deficits or excess based on the hair going back 8 months. So different matrices tell us can give us different insights over, what they've consumed at different times.
Great. Yeah. And that speaks more to what's conserved from individual to individual than to the precision nutrition question of do
Yes.
different things have different impacts on different people. And I think that feeds into a question that just came in, but that I think is relevant, is can you see variation in response varying by ethnicity or other demographic characteristics?
I think we're certainly seeing, differences between men and women. The studies and the numbers we have aren't quite large enough to sort of see if we're getting ethnic differences. I suspect we will. I think, it is known that there are certain populations that are much more susceptible to, say, diabetes or obesity. And so the response to foods and certain types of foods must be either amplified or diminished. I mean, there's certainly, obviously, different populations and their tolerance to alcohol and milk is well known. And again, that leads to what people eat or don't eat. There's also, SNPs that are associated with, cilantro tastes like soap, or people who are super tasters who can taste things that none of us can even detect. So those things obviously affect what people will eat. And some of those clearly are genetic. Some of them do fall out according to ethnicity. So super tasters tend to be mostly women, tend to be mostly of East Asian ancestry. So those are things that affect how people will eat and obviously what their preferences for diet will be. And then in terms of their metabolism, yeah, I mean, we do see some of that, but I just say we just haven't seen that in terms enough population numbers to say, are we going to see this in metabolomics?
So much data, so many fun things you could do with it, with enough time and funding, right?
Yes.
Lots of kudos coming in. I couldn't agree more. Thanks, everybody. Here's a question about validation of these methods.
Sure. So all the methods that I've described are ISO 17025 or 15189 approved. We had to go through this process about 3 years ago. And part of it was because some of the companies that wanted to work with us insisted on it, but it also helped us. And we've really, I guess, become advocates for other labs to try and go through this ISO standardization, because it really helps with obviously the validation, consistency, reproducibility. It also helps with porting these things to other groups, other centers across North America and Europe. So yeah, we go through a lot of validation, a lot of testing to make sure these things are really accurate.
A question about the literature mining. What foods are included in that? Is it USDA foundational foods, legacy foods, broader than that?
Yeah, so the Food DB has, as I say, about 730 foundational foods, but it's a larger number than the USDA's list. So it includes some pretty, exotic game. It'll have a wide variety of fruits that I've never heard of, vegetables that I've never seen before, but which apparently fall into a number of commonly consumed ones in different ethnic groups. It actually has been, we're coordinating with the Periodic Table of Food Initiative, or PTFI, and that is sort of being worked on in the background to make Food DB an integral part of the PTFI study. And this one is a more formal initiative that was started by the Rockefeller, but it's now, I think, being funded by the American Heart Association and several other groups. But the idea is to get foods from every country or continent, and it's about 1,000 that they've got a list for right now. And it's pretty diverse.
So it was just called to my attention that we are past the hour by a full minute, and I was having so much fun I didn't even notice. So there are more questions. People who have questions that we haven't been able to get to, if you want to email them to od@nih.gov, we will try to get back to you. For everyone who's still on, yes, Dr. Wishart, you can see that he's a big fan of transparency and open access to data, and he has given us permission to share his slides. So we will be doing that. Please send an email if you're within NIH. Ask for an invitation to the ODS Seminars Teams site. They will be up there. And just, Dave, thank you so much. This was absolutely fabulous.
Thanks so much, Barbara. Lots of fun. I really enjoyed it, and happy to answer more questions as people want to send them to me.
Yeah, I will forward them to you, and as time allows, would love to know the answers. So thank you, thank you, thank you.
You're welcome.
See you all next month for the next month's seminar, Dr. Round. And see you soon, Dave.
So long.
The speakers disappear.